Hi there. In this video, I'll talk about DNA replication and packaging and segregation, including binary fission and mitosis and meiosis. First, a bit about the nucleic acid polymerases, all of which are related evolutionarily. So there are the DNA-dependent DNA polymerases, though that is DNA polymerase, although it uses RNA primers in vivo, but you can also use DNA primers in vitro. Next are the DNA-dependent RNA polymerases, and we usually just shorten that to RNA polymerase. And that's RNA pol 1, RNA pol 2, pol 3, and there's also a pol 4 and pol 5, all related evolutionarily. Then there are the RNA-dependent DNA polymerases, and this is primarily a reverse transcriptase, but also some transposons and telomerase have reverse transcriptase activity. And then there are the DNA-dependent RNA polymerases, primarily RNA replicase that is used by RNA viruses. So back in the RNA world, there were primarily RNA-dependent RNA polymerases, which still exist in some RNA viruses. Then these one line evolved into the RNA-dependent DNA polymerases. These are the reverse transcriptases. And then there are the DNA-dependent RNA polymerases, which are just simply, we call them pol 1, pol 2, etc. And then DNA-dependent DNA polymerases for replicating DNA. And all of these last three are primarily in the DNA and RNA protein world. In a previous lecture, I talked about DNA structure, and here is that slide again, just to show you that DNA has a 5' phosphate end, that is on the 5' carbon of the sugar, and going down to the 3' end on one strand, and from 5' to 3', where you have a hydroxyl on the 3' carbon of the deoxyribose. So you have the anti-parallel strands here. One can be built up, and then the other one is the template. Polymerization of DNA always occurs in the 5' to 3' direction. That is, the new bases are added to the 3' end using the opposite strand as a template. And in each case, the nucleotides have to be nucleoside triphosphates. So you have to have deoxyribo T, deoxyribo A, etc. being added. During the process, then, only the initial phosphate is used to join into the backbone of the DNA, and the other two phosphates are ejected as what's called pyrophosphate. New strands of DNA are added by a process known as semi-conservative replication, in which one of the strands serves as the template for the other new strand, and then the other strand in the original parent strand serves as a template for the other strand for this other molecule. So each of the new molecules has one old strand and one new strand. Polymerization starts at an origin of replication. Usually it's high in AT because that can be melted out more easily than a high GC region. And it starts in the middle of the molecule, not usually at the ends. And so you have a DNA polymerase starting from the replication bubble, what becomes a replication bubble in the origin of replication, and then proceeding in the 5' to 3' direction, while another DNA polymerase starts on the other strand to copy it in the 5' to 3' direction. The other strand on each one of these can only be copied in the 5' to 3' direction, so this creates a problem in that the DNA polymerase has to be popped into that open area from time to time, and it's done so in a staggered way so that a small piece is added, and then the DNA polymerase pops back further and starts another polymerization back further, and continues that by moving backwards on that strand and doing replication, synthesizing in the 5' to 3' direction. This process creates a series of short fragments known as Okazaki fragments after the person who discovered them. And they're knitted together or joined together by a different enzyme, DNA ligase, which seals each of those together into a longer strand. DNA polymerases are among a class of DNA-dependent DNA polymerases that require RNA primers to start replication, and this goes back to their evolutionary origins as nucleic acid polymerases. So here's one of the forks, replication forks. Here's the 5' to 3' direction, using the parent duplex and making a new strand on this side, which is known as the leading strand. It's called leading because the lagging strand has to put the DNA polymerase back further each time to add these small little Okazaki fragments, which eventually are joined together as a longer piece and joined together by DNA ligase. And you can see the short RNA primers here as well. So there's actually an enzyme that creates those short primers, and then the DNA polymerase can come in and start producing the DNA, and then there's an overreading of that, displacing the RNA primer in each case, and eventually joining the DNA pieces together with DNA ligase. Here's a slightly different view showing a little more detail of that process. Here's the DNA polymerase that's literally clamped around the template and building a new strand of DNA on the leading strand. And then here's the lagging strand here. This is actually going in the opposite direction, and it flips back from time to time to start a new strand, a new fragment. There are also proteins that zip apart the DNA to denature the DNA so that it's available for the DNA polymerase and the primase. Also, there are other proteins here, dark, which are DNA binding proteins, which stabilize the single strand of DNA. So these are single-stranded binding proteins. And then there are enzymes also that untwist the DNA as the polymerase goes down the line, so that the torsion can be released from time to time, so it can unwind that DNA. And here are some of those molecules, including topoisomerases, which cut and rebind the DNA to release some of the pressure as the DNA is unwound. And then the helicase and single-stranded binding proteins here unwind the DNA at the point of where the DNA polymerase is coming in, so they denature the two strands of DNA. Then the primosome creates those little RNA primers, and then ligase eventually seals the Okazaki fragments together, and the red molecules here are the DNA polymerases moving down. Here are two other diagrams of the same process, showing that the polymerases actually associate with one another in this process, in that replication fork. So here are the single-stranded binding proteins, the helicase, the primosome that creates the RNA primer, and there are the DNA polymerases. There's DNA ligase sealing the ends, and the topoisomerase here. So as this one's moving down this way, this one has to wait and flip back from time to time. And it does so in this fashion down here, where this strand is being built and it displaces the other strand. It actually folds out for a time until the primosome can fit in there and make the primer, and then the DNA polymerase flips back to that point and starts in the 5' to 3' direction, synthesizing a new strand from that template. So it's been called, this lagging strand has been called sort of a ratchet system, where it polymerizes and then releases, reattaches, and then polymerizes again. And this process is proceeding rapidly at about 50 nucleotides per second. And at this speed, then, E. coli can replicate its chromosome once every 20 minutes, and some of your cells can replicate their chromosomes once every 24 hours. Without these speeds, then you would not have these organisms evolving the way that they did. So next I want to talk about how chromosomes are separated in cells, but before I do that, I need to talk a bit about definitions and nomenclature. So the C value is the normal haploid amount of DNA, or in bacteria, just the normal amount of DNA. So 1C, or just C, is the haploid amount, 2C is the diploid amount, 3C is the triploid amount, etc. And then multiples of this, we just call the C as being polyploid. N, on the other hand, stands for the number of chromosomes. So in the human genome, 1N, a haploid cell, has 23 chromosomes, and 2N has 46 chromosomes. In mouse, on the other hand, the haploid number is 20 chromosomes per haploid nucleus, and in broadbean, 1N equals 6 chromosomes. So remember those values, because they can change. They can be different. They differ from one another. And as I'll explain later, that a haploid number of chromosomes doesn't mean that you have a haploid amount of DNA. So these, as I say, these can differ one from another. The cell cycle in bacteria and archaea is fairly simple. If we start at a 1C level over here in what's called the G1 or GAP1 phase, the cells can be spores. They can rest for quite a while and then come back. Some cells actually differentiate, so they may differentiate as well. And then you go into the S phase, or the synthesis phase, to make more DNA, replicating the chromosome, then go from a 1C level to a 2C level. And then there's a second gap stage where the cells are at a 2C level, essentially. And then there's binary fission, and each one of those cells then becomes a 1C cell in terms of the amount of DNA. If the bacteria have a single chromosome, then they would also be at a 1N number, but some bacteria have multiple chromosomes, up to 5 in some bacteria. And so the number of chromosomes could vary. Here is an electron micrograph of an E. coli where all of its DNA has been released from the cell. And it has a 4.6 megabase, million base pair genome. And you can see all that DNA has to fit inside of that cell. These photographs are a little bit misleading because the DNA is actually thinner than what is shown here. The reason you can see the DNA at all is it's been coated first with cytochrome C usually, and then with platinum, palladium, a metal that will deflect the electrons in the electron microscope so that it makes the chromosomes visible. So these chromosomes are actually fatter than they actually are, just to make them visible, since they have a coating of some proteins as well as some metals around them. Still, it's a lot of DNA to pack into one of those tiny little cells. This is a diagram of the replication of a circular chromosome, such as that of E. coli. So there's a circular DNA, and it starts replication at an origin of replication, a specific sequence that is recognized by some proteins to separate the strands, and then the DNA polymerase begins on the leading strand, going around in one direction, copying one of the strands, and then a lagging strand on the other side is copying the opposite strand, ultimately winding up with two strands for each of the new duplex molecules, and each one has one old strand indicated by the thinner line and one new strand indicated by the thicker line. This type of replication was shown in I think the 60s by John Cairns, showing that in the middle of replication, the DNA took on what's called a theta form because it looked like a theta, the Greek symbol theta. And this was caught then in the middle of replication where about half of the molecule had been replicated. So you can see the replication forks in two places, and then the newly replicated DNA folded out on both sides, and then the DNA that's yet to be replicated at the bottom. Another form of replication that some viruses, viroids, and even some eukaryotic sequences use is called rolling circle replication. So replication starts at one point where one of the strands is nicked at a specific location, followed by initiation of replication by the DNA polymerase at the site of the nick. So that becomes the leading strand, and then there's a single-stranded piece of DNA that is displaced. And that becomes the lagging strand. So a DNA polymerase pops on, and then by a ratcheting system moves back further to initiate replication as that single-stranded DNA becomes available for replication. All the time the leading strand is proceeding to replicate the opposite strand and pushing out that single strand. Ultimately, multiple copies of the genome, which was circular and now is linear, are produced by this mechanism, so that you have concatemers, essentially copies of the entire circle repeated over and over and over, head to tail on that linear piece of chromosome. Usually during the replication process, the cell is continuing to grow in preparation for division. In the case of archaea and bacteria and some eukaryotes, this occurs by binary fission, and that starts with the chromosome there inside the cell, and it's normally attached to the cell membrane by specific proteins. During replication then, some of those proteins add to a similar site on the chromosome that has started replication, and the cell begins lengthening, and it usually lengthens from the middle of the cell. So you're getting membrane and cell wall added to the central part of that cell, which causes those chromosomes to begin separating by moving towards the poles. As the cell walls and cell membranes continue to move outward, the chromosomes are pulled apart and ultimately wind up at the poles. The cell wall and membrane begin to pucker inward in the center of the cell, and then the cells divide by binary fission. Then this process repeats over and over again. So this is the standard and usual mode of duplication of the chromosomes and separation of the two cells. So here is that same process shown a little more schematically, because I want to show some other possibilities. This is binary fission, where you have a bacterium at a 1C level. The chromosomes duplicate. Now you have a 2C cell that's ready to divide. It ultimately divides, and you have two 1C cells. However, many bacteria become polyploid, so they begin to replicate their chromosomes from a 1C level to a 2C level, 4C, 8C, etc., and become polyploid. Normally they will do this when they need a particular gene to be amplified. That is, they have to have multiple copies of a certain gene because they need a certain gene product. So this is actually fairly common in bacteria. Many bacteria also can undergo what's called extrachromosomal amplification, where they can loop out particular portions of their genome. It can actually replicate on its own, and then it can reintegrate into the chromosome. This happens often when bacterial cells are stressed in an attempt to deal with the stressor, whether it's a toxin, lack of nutrients, things like that. It'll pop out a bunch of genes and then replicate those, hopefully mutating enough so that it can deal with the stressor, and then reintegrate those mutant copies back into the genome once that particular cell line becomes successful. If not, they just die. So it's a process that does rely on Mendelian processes and Darwinian evolution. But when you actually see it in action, it seems more like Lamarckian evolution, but it turns out it's not. It's simply another adaptation that has evolved in bacteria to deal with highly stressful environmental situations. And then there are plasmids. These are extrachromosomal, autonomously replicating pieces of DNA that carry particular genes that can be moved from one cell to another, one species to another, and carry particular genes necessary for many different functions, actually, for heavy metal resistance, for antibiotic resistance, for invading cells, etc. So these are some of the things that carry antibiotic resistance in human pathogenic organisms that can, so you can get rapid evolution, rapid adaptation by bacteria to survive certain antibiotics by obtaining the plasmid that encodes a gene and a gene product that will allow it to live when it's exposed to those antibiotics. So some of these can cause rapid adaptation and rapid evolution in bacteria particularly. But plasmids occur in other organisms as well, including archaea and fungi and some other eukaryotic species. So if we go back to the cell cycle, these particular adaptations and changes have to fit within this cell cycle. So here's the normal cell cycle. But if you have endoreduplication here, you can skip the G2 phase, go directly back into the G1 phase, and skip fission, skip division. And so you can go through the S phase over and over and over without cell division. And it may seem, yeah, you can do this on paper, what's actually happening. You can actually detect some of these key points at these lines, and certain genes and gene products allow the skipping of some of the stages of the life cycle under certain circumstances, certain environmental circumstances. And for the amplification part, all the cells do is go through the G1 and then into the S phase, where at a certain point in the S phase, certain pieces of DNA are excised from the chromosome and start replicating on their own and amplifying those particular sequences over and over again. So here's the cell cycle for eukaryotes. Now, eukaryotes have mitosis, at least most of them do, and they have a meiotic cell division cycle as well. And so there are certain parts of the cell cycle that are more elaborate than they are in bacteria. Plus, many eukaryotes, but not all, are primarily diploid organisms and have shorter haploid parts of their life cycle. So that's a difference as well. So if we start in G1 with a diploid cell 2C, And that's a normal condition for most large organisms like humans and trees and things like that. They have a resting part that they're not actually resting in most cases. Or there can be differentiation or apoptosis, cell death. But if we continue around G1, go into the S phase, then you have the nucleus going from a diploid to a tetraploid stage. So you have 4 times the amount of DNA as a haploid. And in G2, then you're a tetraploid already, and then go into mitosis, prophase, prometaphase, metaphase, anaphase, telophase, and then K is for cytokinesis. So then you're reducing the number of chromosomes and reducing the amount of DNA in each nucleus by half to return to the diploid nucleus. Now, in organisms that are mainly haploid, such as fungi and some protists, you start from 1C, build up to 2C in the S phase, and then drop down, back down into the 1C stage, 1C amount after mitosis. During the S phase, during replication, eukaryotes have to do things a little bit differently. One is that they have very large chromosomes, so they have to have many origins of replication. The second thing is most of their chromosomes are linear, except in a few eukaryotes where they're circular. But having a linear chromosome presents some problems. And the problem comes not on the leading strand, but on the lagging strand. So at the time that you get down to this very end of the chromosome, you can't fit the polymerase onto the end and copy that last little bit. So if that were to occur over and over again, the chromosomes would get shorter and shorter and shorter, which would be a situation that would be lethal ultimately. So during replication, early in development in organisms, telomeres are added to the ends of each one of the chromosome arms. So here, these white regions of each one of the chromosomes on the ends, those are the telomeres. And telomeres are added by a protein called telomerase, which is a reverse transcriptase. So each of you in your genomes have telomerases, which are reverse transcriptases, which use an RNA primer to make a DNA copy. So along the way here, there's the telomerase and the RNA primer within that, and it copies it over, and then it shifts over because it's self-complementary and keeps adding these repetitive sequences to the ends, so that that during the ensuing replication cycles, the chromosomes do get a bit shorter, but they're only eating into the telomere region and not into the gene regions. To finish off the telomeres, DNA polymerase gets involved also. So here is the leading strand, the lagging strand with the Okazaki fragment still there. And then the telomeres are being added on here. Ultimately, that loops around, and then DNA polymerase comes in and finishes off that entire piece. And finally, the last nicks are connected together with DNA ligase. So that completes the telomere. So this was a very important innovation that allowed these long linear chromosomes in eukaryotes to evolve. Otherwise, they would not have been possible. And the reverse transcriptase probably came from virus sequences or transposons or some other genetic element. So not all viruses are bad. Some have led to a lot of evolutionary innovation in larger organisms. So chromosomes have gotten so large that there's a great deal of organization necessary to tie them together, to control them, to pack them in certain areas, to pack them during mitosis, to open and close the strands when the sequences have to be expressed or not expressed. So here's a little bit about the chromosome structure. If you look at a chromosome and you cut across it in one place, it would look like this with all these loops of DNA coming out. And the loops are about 300 nanometers in length, and they look like this coming out from the chromosome. And then if you take each one of those little strands there and look at that, it's actually DNA bundled up with histones into a 30 nanometer strand. And then if you take out histone 1, then you can stretch that out further. And when you look at this structure, it's been called beads on a string. So it looks like little beads with strings holding it together. And about 140 nucleotides are wrapped around each histone. And then if you strip the histones away, then you have the naked DNA, which is about 2 nanometers in diameter. And if you took the DNA from only one of your cells and stretched all those pieces end to end, they'd be about 2 meters or about 6 feet long. So it's a lot of DNA in each cell. Because the chromosomes and the amount of DNA is so large, only one origin of replication would take years to replicate the entire human genome. So there are multiple origins of replication on each chromosome, and they replicate at different times in the S phase of the cell cycle. So in white here are regions that replicate early, and then shades of gray indicate regions that are replicating later and later, and the dark regions are the last regions to replicate. Replication of all the chromosomes in the human genome at the fastest rate takes about 24 hours. And on each replicating strand, they're replicating at about 50 nucleotides per second. So we have 23 sets of chromosomes. Other eukaryotes have anywhere from 1 to 630 chromosomes in their haploid set. And so each one of those is going to take quite a while to replicate. Usually for the very smallest genomes, several hours. And to the very largest genomes, it can take several days to replicate the entire genome for one replication cycle and one cell cycle. Here are a few more details on mitosis specifically within the cell cycle. So here at interphase at G1, the nucleus is at a 2C level for most eukaryotes. And then in interphase in G2, it's replicated its chromosomes, so now it's at a 4C level, and it has, instead of one nucleolus, it has 2 nucleoli, and those are where the ribosomes are being assembled. Then into prophase, where the chromosomes begin to condense, and then prometaphase, where the chromosomes have condensed and the microtubules begin to grow and connect the chromosomes to the poles. And then metaphase is where the microtubules have pushed the chromosomes to the metaphase plate in the middle of the nucleus, or the middle of the cell. And then anaphase, the chromosomes are pulled apart by the microtubules, and there's another set of microtubules that is pushing the cell poles apart. And then in telophase, the cells are being pushed apart a little further, and the nuclei are reforming, and the chromosomes are decondensing. And finally, you get cytokinesis, where the cells are completely separated, and they're back into the G1 phase, where they're diploid cells. This is just a picture showing some of the important molecules that are involved in this process. So first are the Golgi, and those are the organelles that are responsible for forming a lot of the modifications on proteins that will wind up on membranes, including the nuclear membranes. This next one is for beta-tubulin, and that forms the spindle or the microtubules that pull the chromosomes apart to the poles and also push the cells apart. The DNA there is stained with DAPI stain, which stains blue, and then the last picture is a composite of all three showing the close association between those molecules and organelles. This is a diagrammatic representation of what's going on with the chromosomes during this time, separation during mitosis. So initially in the S phase, you have these homologous chromosomes, one coming from each parent, duplicating. So now you have these chromosomes that have duplicated, and you have two sister chromatids rather than a single chromatid or chromosome. The kinetochore microtubules then during prometaphase and metaphase are attached to opposite poles of that cell, and each one is connected to either one of the sister chromatids or the others at the kinetochore or centromere. The centromere is a specific part of the chromosome, and the kinetochore are the proteins. It's a protein complex that attaches to those sites and attaches to the microtubules. During anaphase, then, the sister chromatids are pulled apart, becoming chromosomes again. So there's a sort of a change in the terminology for those chromosomes and chromatids. And then in each cell, then, you have a pair of the copies of the pair of the original homologous chromosomes for separation into two different cells. Mitosis differs among different types of organisms. So in animals, normally there is what's called astromitosis. So you have these asters formed, or centrioles formed, at the poles of the cells, which become microtubule organizing centers for polymerization of the polar microtubules, which go from pole to pole, and the kinetochore microtubules that go from the pole to the kinetochore of the chromosome. And then there's anastromitosis, where there are no centrioles, no asters formed. But there's a light staining region in the cell that is also a microtubule organizing center. Again, it's a site for polymerization of the polar microtubules and the kinetochore microtubules. Both the centriole, or the aster, which is an MTOC, and the MTOC in anastral mitoses, primarily in plants, are filled with monomers of tubulin, alpha, beta, and gamma tubulin. For most organisms, the kinetochore microtubules attach the chromosome to the microtubule organizing center. But in a few plants and animals, they have holocentric chromosomes. So the microtubules essentially attach to multiple sites on those chromosomes and attach those to the MTOCs for preparation of mitosis. So this is just another example of the variation and diversity that you see in various functions during evolution of certain organisms. But there are many other variations on segregation of the chromosomes. So in most dinoflagellates, which are protists, eukaryotic protists, within the nucleus, and I'm only showing the nucleus here, the chromosomes are attached to the nuclear membrane by certain proteins, which are probably sort of like kinetochore proteins, but not exactly. And then the polar microtubules go through the nucleus, and they don't go into the nucleus. They just form these little tunnels through the nucleus itself. So the nuclear membrane is fully intact the whole time. It never disintegrates. During mitosis, then, the chromosomes begin to move to each pole of the nucleus, and the microtubules and the cells tend to expand. The nucleus elongates and eventually divides into the two nuclei carrying each new nucleus carrying one set of the chromosomes. In some ways, this looks analogous to a bacterial or archaeal cell, when it's dividing without a nucleus. Remember, over here with the dinoflagellates, I'm only concentrating on the nucleus. Here with bacteria and archaea, I'm talking about the entire cell, but it resembles binary fission, but in this case, fission or division of the nucleus itself, and the cell outside of that is also dividing at the same time. So evolutionarily, these two sort of do tie together in a way. For some dinoflagellates, they have centrioles and asters, and the chromosomes are attached through a protein to the kinetochore microtubules. So you have both polar microtubules and you have kinetochore microtubules in these. And during division of the nucleus, then during mitosis, this nuclear membrane never breaks down, but you essentially get fission of that nucleus into two new nuclei, each carrying one set of the chromosomes. In ascomycete fungi, such as Saccharomyces cerevisiae, which is the yeast that you put in your bread, in your beer, in your wine, etc., they also have a mitosis where the nuclear membrane never breaks down. And in addition, they have microtubules both outside of the nucleus, and they have microtubules that form inside of the nucleus. So in this case, you have polar and kinetochore microtubules that form inside, and then actually you have microtubules outside of the nucleus, also positioning the nucleus. But essentially, this also sort of looks like that in bacteria, but much more elaborate in that now you have microtubules rather than just simple attachment to the nuclear membrane to separate those two nuclei. In basidiomycete fungi, it's even a little more bizarre in that the initial part of mitosis, the chromosomes are actually pulled out of the nucleus, and so the nuclear membrane eventually breaks down, but the chromosomes are pulled out of the nucleus by microtubules, and then microtubules form, including polar microtubules and kinetochore microtubules, and a new nuclear membrane forms around each one of those new nuclei. And so this is sort of caught at that point in time when the chromosomes have already separated and the new nuclear membranes are beginning to form around those chromosomes. So this diagram shows some of those other details as well. So here is in ascomycete fungi or ascomycetus yeasts, where you have the microtubules beginning to form. The little blue dot are the chromosomes, which never can be seen as individual chromosomes. It's just a bundle of chromosomes. Microtubules form both outside and inside the nucleus. Then there's been duplication of the DNA. Those two are moved to the poles of the nucleus by microtubules. Other microtubules are starting to push out this bud here, and then you get elongation of both the bud of the cell and the nucleus. Finally, separation of the nuclei and separation of the two cells by cytokinesis. In basidiomycetes, down here, basidiomycetus yeasts, you have a similar situation at the beginning. You have the chromosomes duplicating, microtubules forming outside of the nucleus, but the microtubules outside of the nucleus are pushing out the bud, and they rip the chromosomes out of the nucleus, and the nucleus eventually, the nuclear membrane, disintegrates over in the corner. New microtubules form, pushing the two chromosome sets apart, as well as pushing the bud further out. And then this one shows the new nuclear membranes beginning to form around the chromosomes, ultimately forming a new nucleus and separation of the two daughter cells. In plants which have anastral mitosis and firm cell walls, then mitosis occurs in a slightly different way. On top are shown some photographs or some micrographs of some micrographs actually that were taken by a graduate student that I knew. So these are fairly good, although she had to do it in a cold room and over a period of like 12 hours or so to look at these. Anyway, so you have the cell here beginning to go into prophase, prometaphase, and then metaphase, where the chromosomes are lined up in the middle of the cell, but the microtubules look more or less parallel to one another. And then you go through anaphase and finally telophase, reformation of the nuclear membrane. So in this case, there still are polar microtubules pushing the cell poles apart. The cells will expand. Mainly the cell walls are softened and the cells elongate somewhat. So there are polar microtubules and then the kinetochore microtubules. And then for cytokinesis, a new set of microtubules forms near the metaphase plate, that is the middle of the cell. And instead of carrying chromosomes back and forth or pushing the cell, these ones contain or carry cellulose synthase molecules, enzymes, and those will lay down cellulose at that middle plate of the cell, building up new cell wall material to separate those two cells. So cytokinesis in plants is very different than cytokinesis in animal and protist cells, by and large. So if we make sense of these different forms in an evolutionary sequence, then you can look at bacterial fission as being the simplest form of duplication of the chromosomes and separation of the cells. Then you have mitosis with intact nuclear membranes, and then add on meiosis a little later. And then mitosis with a donut-like nuclear membrane plus meiosis. And then mitosis with partial dissociation of the nuclear membrane with meiosis. And then finally mitosis with disintegration and later reformation of the nuclear membrane and plus meiosis. So if we look at these on a phylogenetic tree, so here's a tree with a root here, there's a bacteria and archaea, and then all of these are eukaryotes up here, these are protists here, plants there, amoeba there, and animals and fungi up here. So down here we have binary fission in bacteria and archaea. It also occurs in some primitive protists also, so it may extend up a little bit into the eukaryotes. And then we have mitosis and meiosis evolving in the eukaryotes. Now, meiosis is pretty much a recombination event followed by a mitotic-like division. So it's essentially taking mitosis, adding a form of recombination onto it to increase gene diversity and get rid of mutant genes, and then a division after that. So it was not a completely new innovation. It was taking a division form and then adding a few things to that. So next you add on organisms over here, the protists, where most of them have a mitotic division where the nuclear membrane does not break down during mitosis and cell division. But one type has something that looks like anastral mitosis, and the other one clearly has astral mitosis, but without nuclear membrane breakdown. And then there's this odd group up here that are the fungi, where in one case you have the nuclear membrane not breaking down, but you have microtubules inside the nucleus as well as microtubules outside the nucleus. And in the other case with the basidiomycetes, you do have breakdown, or not breakdown, but the chromosomes are ripped out of the nucleus, and then a new nucleus forms around each one of the two sets of new chromosomes. So that one is unique among organisms as far as I am aware. Next, there's this group, the animals, which have astromitosis with complete disintegration of the nucleus and then reformation of the nucleus during telophase. Mitosis in amoebozoans, is very similar to that in animals. It is astro with disintegration and reformation of the nuclear membrane. And finally, in plants, Archaeplastida, you have anastromitosis with disintegration and reformation of the nuclear membrane. So there's quite a bit of diversity in mitosis and cell division and chromosome division among organisms, but you can see a general pattern of evolution in that it's becoming more and more complex as time goes on. Okay, so that's the end of this video on DNA replication and segregation of the chromosomes. I'll talk to you later.